All right, well, you guys can notice I'm very short, so we're gonna have to bring this way down or bring a microphone. So I'll try and stay next. I like to walk around, but I'll try and stay over here. Um, First, thank you for having me. When I walked back into the building, it was like coming home. I lived in this building for about 6 years. I can tell you the best stairwells to cry in, the best places to grab coffee, and whatnot. But it's been really nice to be back in biodesign. I'll start off at the ending, essentially, being an MSL right now. Does anyone know what a molecular science liaison is? Raise of hands. Besides Dr. Monk, you can't answer this one. All right, well, I had no idea either. A molecular science liaison or medical science liaison is a really unique position. They're basically part of PharmDs or any industry where you're selling a drug, a diagnostic platform, whatnot. And it's essentially the dream job that I never knew existed. I get to work basically in this little fine line between academics, so we work on research projects, I work with ASU, I work with Mayo Clinic and whatnot, and I work with clinicians. So at Caris, what they essentially do is they assess all solid tumors for different biomarkers that could be targeted by approved drugs. And so that science part that I got that PhD to be able to do is essentially liaise. I bridge the science with the clinicians, and I'm able to talk about everything in between and basically manage research projects. And so when I was invited to do this, it was really made me have to think about how I got here. And to be completely honest, I had a pretty non-traditional route. I come from a Hispanic Filipino family, whereas many of you can imagine, I was very much pushed for school education is first. Neither of my parents went to college, and I'm the first of my dad's kids to actually graduate from college. With that said, there was zero direction. And so even going to college was absolutely new for me. The one thing is I have to thank my mom, because she was the one looking at the letters and like, oh, like, why don't you apply to this college? What about this one? And I was like, okay, how do you even apply to college? And so thinking about where I'm at now, from where I started, is really interesting. I think it's important to talk because it's not just, this is my journey, but I think a lot of people can share a lot of the similar trends. But thinking about it, one of the things, the three common themes with being where I'm at now is mentorship, networking, and a whole lot of grit. One thing my mom always said is, Peaches, you are so stubborn. But it ended up working out for me. So I'm going to start essentially fast forwarding to Whittier College. This is where I did my bachelor's degree. It's a small liberal arts school in California, and it's essentially the complete opposite of ASU. The largest class is about 30 students is the largest class. And so when I came here and I walked into my immunology course and there's like 300, I was like, oh my God, like this is crazy. So needless to say, I was relatively spoiled, being at a small school where you can just walk up to your professors, they have an open door policy, and so on and so forth. Now, going to Whittier, like most people that I grew up with, I was pre-med. And this essentially is because my background with my family is the three successful careers at the time was you're a medical doctor, you're a lawyer, or if you ask my nana, you're a secretary at a bank. And so those are my options. And so going into undergrad, I was like, you know what? I love science. I geek out over it. I'm going to start doing the pre-med courses. And this worked out for me because I loved it. And at a liberal arts school, I got a lot of practice writing in pretty much every topic, even math, which was weird. You had to do essays in math. But you know what? It really helped me hone in those skills. So I was a major in biology. I minored in kinesiology. And as I moved up in my courses, I had a professor ask, hey, Peach, you want to spend the research doing ecology research? I was like, what is that? What is ecology research? She's like, you're going to go out into the National Tahunga Forest. We're going to measure the development of new species in proximity to the water. And I was like, why would we do that? Like, what in the world? But she was trying to see, like, how regrowth of species was affected by proximity to the water due to all the fires that have happened. I saw it as getting paid for the summer to go hang out in the national forest, and I was like, yeah, let's do it. So it was great. We got papers out of it, presentations, whatnot. But I imagine, I said, if this is grad school, if this is research is, I don't want to do it. Like, there's no way. My biggest concern was not stepping on a snake out there. Needless to say, I kept moving up in my courses, and I got into some of the more senior classes, which were immunology and oncology. Now, there were 4 biology professors at this college, one of them being an immunologist. And so she was teaching those courses, and I got invited to do bench research. And so I was like, all right, let's give this research thing another go. And I was looking at C. elegans. Marcos, you'll appreciate this, because I was looking at C. elegans, I was under the microscope, and I geeked out over it. I was doing an experiment, essentially, where we were feeding these C. elegans pathogens, measuring their death and the toxicity of them. But the unique thing is this called for being in the lab every 4 hours to count how many of these worms died. Yeah, I was like, what do you mean every 4 hours? So, essentially you're sleeping 2 because you have to get up and go to the lab, take the elevator up and all this stuff. And it was over about 80 hours. So it's a lot of time of no sleep and a lot of head nods. I know you guys know what I'm talking about. So it was hard, but you know what? I loved it. I didn't complain. And as soon as I got that last set of data and could go back and analyze it and look at the results, I was excited. The 5 a.m. trips to the Tahunga Forest, not so much. But doing this, I knew I would love doing bench research. The problem is, it's pre-med. I started taking these courses and getting invited to do bench research around the end of my junior year, the beginning of my senior year. And to be completely honest with everyone in this room, slightly embarrassing, I didn't know what graduate school was. No one in my family had gone past high school, really. And so I was kind of learning everything as I was going along. And thankfully, I had some phenomenal mentors at Whittier College. One of them, the immunologist, Dr. Sylvia Vitrone. She took me under her wing, and she's the one who got me started with doing research, starting to look at how to write papers, mentored me over the summers. And, I had that struggle. I'm pre-med. I want to become a doctor, make my family happy. But my heart just wasn't in it anymore. But how do you have that conversation with very traditional parents that are expecting you to become a doctor because that is how you're going to be successful? And so, around this time, both my best friend and my grandma had cancer, lung cancer, stomach cancer. And so in the background of all of this, I'm kind of dealing with those questions, whatnot, trying to figure out what this cancer thing is, why it works, why it's affecting, both my best friend and my grandma with these things. Being at the time for my grandma, I did hospice care for her. I was in every single doctor's appointment with her, talking to the oncologist, being on the other side of it. And one of the most frustrating parts for me was the absence of answers and the knowledge gap. I was very frustrated that I didn't understand what was happening in their body. And so that stuck with me because at this divot point, essentially getting into my senior year of college, am I going to take the MCAT and apply to medical school, or am I going to do something else? And so while my mentor was having me do research and whatnot, she started talking to me about graduate school and what that really entails. And so because of that, I learned that while I could be an oncologist and be face-to-face with the patient, I could also play a role by being in the background and understanding the disease more and trying to develop or understand it more, find more answers, and hopefully have it go into translational research. And so that's where I was at during this time at Whittier College. I was really torn, but my mom saw it in me. And she said, Peaches, why do you love going to the lab? Why don't you apply for grad school? And it was that, for some of you may be silly, but that was a permission that I needed, culturally and everything in our family, to be able to pursue that dream. And I was lucky enough to have my mom to really recognize that. And so I was like, how do I apply for grad school? For any of you that, most of you have applied, it's It's a long, tedious task, and you're expected to have a lot of research experience. And at this time, I just didn't. And so it led to essentially this gap year where I graduated in 2012 from Rodier College, and my mentor, Sylvie Vitrone, was able to introduce me to a colleague at USC, where I was further able to do another set of round of research under a breast oncologist there. And it was nice. It was a lot of basic science. I was running gels day in, day out. And again, it was just something to bulk up my resume. And at the same time, Kaplan, I don't know if any of you are familiar with that, but they run an international school. And so I applied for the summer and was able to be an English teacher and teach students from all around the world how to speak English. And let me tell you, when I walked into that room, I didn't realize that they wouldn't know a single word. And so I was like, how am I? I didn't know. I was like, they're like, oh, it'll be a range. And I was like, this is not a range. So I had to be creative on the spot and figure out, how am I going to teach this group of 15 students how to speak English? And so even describing baseball and whatnot, I got creative. We were doing drawings, we were, cutting things out and whatnot. But the main thing about this is that it taught me that I love to teach, that I love to be in that mentorship role, and that you have to be creative, and everyone has a different learning style. That's my biggest thing. They're all learn differently, so you, as a teacher, you have to adapt to that in order to get the message across. And so this is what I spent my first year gap doing, is essentially trying to get more research experience and also having some experience as a teacher. I was also an adjunct at Whittier. I got to do like their microbiome labs and whatnot. So I stood busy trying to make myself just a better candidate. Now, that same mentor, she's like, all right, Peach, you're coming up over a year. What are we going to do next? To be completely transparent, no one likes to talk about the bad. But during my time at USC, during that year, I did apply to grad school. I got a couple interviews, and I didn't get in. And I was devastated because I didn't know what I wanted to do after. I remember sitting there and wondering, what other career would I want to do outside of science? And I said, well, I've always wanted to work at SeaWorld and like train with the dolphins, right? But I was like, it was like maybe marine biology. And then I was like, actually, like you're terrified of the ocean, so that would not work for you. So I was like, maybe I'll stick to science. So we talked, and at the time, the NIH did a funding for a post-bac program. And so it's basically a year of getting your feet wet as like a graduate student. You get submerged in the life of a grad student, and it's fully funded by the NIH. And so I applied. I got into ASU, as you can guess, as well as Santa Barbara. And my mentor told me, pick a school where if you were to stay in the lab that you got into, you would want to dedicate your graduate career there. So I looked up ASU, looked up immunologists, and everyone here, and Karen Anderson popped up right away, along with Doug Lake and Joe Blattman. And I was reading about her research, looking at all the website, and I was like, that looks cool. That looks awesome. MD, PhD, she knows what, all this is going through her whole CV. So I applied, and I ended up with Dr. Hogue in the back, and she basically held my hand through that year of immersive training. And I was graciously allowed to be in Dr. Anderson's lab, where the unique thing is I was co-mentored in the beginning, and we was working on a project that was more industry-based. I was also with you, and it was looking at profiling T-cell receptors using this new technology. We know that there's already technology for it, but it was a neat aspect because it wasn't just looking at translational science, but how do you make a new technology? Now, granted, I didn't mention this before, but in my past, when you're growing up, it was basically you do as you're told. You don't question the professors, you don't question the doctors, you kind of just do as, you don't cause waves. And so coming into this institution, remember, Karen, one thing I love about you is you don't hover over people. You're like, all right, you have a very established lab, you had postdocs, grad students, and everything, and you scooched me on in there, And you basically gave me the freedom to ask questions without being told, like, no, you're not allowed to do that. This is how things work. And so coming upon I applied for grad school. I got interviews here and at MD Anderson, got accepted, and I chose to stay here. Not only because I was already working on this really phenomenal project, but I was in a unique environment. In the beginning, I said mentorship was really important, as well as networking. So when I walked into this lab, Karen, I really think everyone should follow your format of how you run a lab. You have your postdocs, and then you have your grad students, and you have your undergrads. And essentially, everyone's mentoring everybody else in this very unique way. And so one thing I noticed about the lab right away, I was the only student from the US, the only US citizen in the lab at the time as a grad student. And that was really critical when I think back, because I was being mentored by individuals from India, Egypt, Mexico, so much more advanced than myself. And a lot of what our colleagues talked about in the past, it's not just about the science. It's really about you growing as a person as well. And I was very lucky to have those people to help me learn soft skills, speaking in front of people, asking those questions why. Why is that person acting weird today? Thinking much more outside of just what you're doing at the bench, which I think, in part played into how I can be in an MSL now today. So I joined Karen's lab in 2015 officially as a grad student, and we've spent 5 years married to one another, is how I like to put it. We had the good, the bad, and everything in between. But I really appreciated her mentorship because she allowed me that freedom to kind of make my own path as a scientist, but still never letting me stray too far away. So with that, I also met two very important individuals as part of my networking, one of them being Karen's postdoc, Benjamin Catchman, and the other being Dr. Blattman's senior student, Louis Shuttle, both of which ended up being my bosses in the future. So funny story though, Ben was the quietest guy ever, and I would just kind of sit next to him and be like, hey, wanting to learn from him. He ended up teaching me how to optimize PCR. That's the first thing he ever did. But I think he was just trying to get rid of me because he's like, here, read this book. And it was like a manual of PCR. But then I would keep coming back and back. And that's one of the things where he's like, I really like that you just wanted to learn. I would give you something, you would take it, and then come back with more questions. You always wanted to know how things worked. And so as I was coming close to graduating in the wonderful year of 2020, again, this room reminds me of just all the COVID tests that we had running in here. One of my last memories is getting a nasal swab in this room. But, 2020, we had a lot of COVID testing. We were able to be part of that initiative. And Karen also allowed me to sneak into the lab and continue my experiments so I can finish up my dissertation. From that, I went on to work at a small startup called Pathogen DX. And lo and behold, the manager there, it was Benjamin Catchman. And so I did my interview process and whatnot, and he got me set up essentially to move right on into that. Now, many of you have probably been told, hey, when you're looking for a postdoc, choose something outside of your field. How many have been told that? To explore something outside of what you normally do. Well, I did. And what you're seeing here is a microarray. I led the food and agricultural testing. So for those of you that don't know, Yuma has most of our lettuce that gets distributed all over. And a lot of it basically gets thrown out because it can't get tested for E. coli and other pathogens fast enough. And so I helped develop this assay that we were able to patent to essentially be able to detect different bacteria and pathogens, Campylobacter, E. coli, Salmonella, and whatnot. So we could use it for testing poultry, beef, and lettuce throughout different industries. And it was really neat, but at this point, I was still doing the bench work and whatnot. I never thought you could get a pipette out of my hand. But as I grew in this role, I was given more senior responsibilities and actually led the team by the time my postdoc was ending. And so at this point, I was analyzing the data, designing the experiments, giving direction to my team of what to do, and that really gave me the first inclination that I would move away from the bench because I thought I was going to die with a pipette in hand, never leaving wet lab work because I just loved it. But this really opened up my mind to that. So as my postdoc was ending, I sent some fillers out to all my friends who graduated previously. It was like, hey, what's everyone doing? Because in academia, a lot of the times, you can become a professor, but what other options are there? Scientific writing, MSL, maybe some other ones out there, but at least while I was here, we didn't get very much exposure to those things. And so, Louis Shuttle, senior PhD in Blattman's lab, he was my mentor for the 4 years, and he would literally run away from me. Every single day I came up to go over experiments because he was on his way out. He didn't want to keep mentoring people, but I was like, look it, buddy. I said, I remember this clearly because it came to that point. I was like, I have to report to Karen. I have to show her experiments. He was doing this project in mice, then I was replicating it in human. And he would run, and he had access to rooms I did not have access to. And the flow cell upstairs, and so he would run, and one day I got him early in the morning, and I was like, look, I am not your friend. I said, but I have to learn from you. And you have the knowledge that I need to complete this project. And he smiled and looked, and that was it. But from there on out, he would meet with me, we'd keep it brief, and I got what I needed to keep learning. But what I didn't know at the time is it was this interaction that really sold a lot of respect and showed the grit that I needed, that we don't have to be friends, we don't have to do these things, but we need to be professional and work together. And so he was the one who texted me and was like, you should apply for this spot. You speak well, you know your science, and I think you'd be really good with people. And so I did, and from 2021 until now, I've been an MSL at Caris. And when I think back to it, this networking that biodesign facilitated for me is what really got me to this spot. Not only Louis, not only Ben, but it was Karen, it was this institute itself, the building. Again, someone had mentioned, you look across, you can see what the other person's doing. I'm nosy. And so one of the things I love to do was look at what other people were doing in their labs and ask questions and see, hey, what experiment are you running? Can I just tag along? Because I want to know how it works. And so that's how I met other people in different labs. And I would say that, the 6 years that I spent here, one of the best parts was the people. Till this day, walking in here, I get to see Marika, Padma, Sandy, Marco, Brenda, everyone that bring back good memories for me. But they all played a really pivotal role in me being able to be in this spot right now. I'll tell you that 3 years ago, even when I graduated, presenting in front of people was terrifying. I had bubble guts the night before, I want to throw up, so much anxiety. But really the amount of presenting, he wasn't even my PI, and from his students, I learned how to put presentations together, how to speak, what to cut in, what's cut out. So having, Karen as my primary mentor, I had so many other mentors that I got to learn from at this institute. We had, what are they called? I can't remember them. CPD meetings, yeah. And so, I know Friday mornings are like, oh God, a CPD meeting. But now I'm like, man, that was awesome because of that exposure and whatnot. So I can't speak for other institutions, but I know that biodesign has played a huge role in my soft skills, in my networking skills. Obviously, it helped me land both of the 2 jobs that I had after a graduate student. And I'm at Caris now, which is actually local. It's probably 15 minutes from here. And again, it's my dream job. So we're looking at different biomarkers in solid tissue tumors. We're able to provide, additional therapy opportunities in addition or outside of chemo, radiation. But mostly, I get paid to study and to learn every day. And so that's the fun part for me. There's something new to learn all the time. And although I may not be an MD, I did get my PhD and make my family very, very proud. But it also really allowed me to understand and value the importance of giving back. And so in this role as well, I'm allowed to volunteer a lot. I'm allowed to come do talks such as this and share. And I get a lot of people reaching out on LinkedIn, which if anyone has and I haven't reached out, it's because I'm terrible at looking at LinkedIn, but I'm happy to share my number. But they're always like, what's an MSL? How do I do it? What do I look for? And I think those conversations are really important to have, because for myself, again, secretary, MD, lawyer. Those were my only options, and there was no guidance. So without that mentorship and the networking and people giving me a chance, I truly wouldn't be here where I am today. So I'm very grateful for Biodesign, the Institute, and the professionals that you have in this building that really pour into their students. And that's it.
Let's see if we can. One question, maybe. Questions? All right, back there. You want to be by the mic, Peaches?
That was a great talk, thank you. I was curious if you had any pre-job recommendations for jobs before getting an MSL job, since I know a lot of the times they require previous work experience.
That's an excellent question, because, again, why being at ASU and networking, even with someone I didn't enjoy working with at the time, but is a phenomenal boss now, really pays off, because he worked with me for 4 years. He knew the role. He is now a director in that role. And like you said, it does usually take a lot of experience to break into an MSL position. But because of that networking prior, I was able to have him vouch and get me at least a foot in the door and then go through the process. So most of the time I say, if you're looking to break into an MSL position and don't really have any of that experience prior, just start networking at those conferences, at the industry conferences, or just reaching out to the current directors and whatnot to get their feedback.
All right, thanks again, Peaches, and thanks to all the morning speakers. Great session.